Now let's talk about how to set up the pipeline. That is to say, how do we get all the files we need to run the core pipeline, aside from the data to be analyzed? Well, we first download the relevant FASTA files from an appropriate online database, such as UCSC. These FASTA files make up the genome of our organism of interest, with each chromosome's sequence stored in a separate FASTA file. We may also need to download the PhyX genome. The files are modified as necessary and then combined. This gives us a single FASTA file which contains the genome of our organism, as well as any other relevant sequences like the PhyX genome. Using this file, we construct an index. We now have an index for our organism's genome which we can align our data against. Let us go into a bit more detail. There are numerous online databases from which you can download genome assemblies. I would recommend the UCSC genome website. Now, this may not be the case for all online databases, but with the UCSC one, there are two ways to download data: via an internet browser or via command line. If you want to do it via an internet browser for the UCSC database, you start by going to the following URL and then scrolling down or using the search function to locate your organism of interest, as well as the assembly version you want to use. Once you've found the assembly you want, click the Sequence Data by Chromosome link. That should take you to a page with a similar URL to this URL on screen, and have an appearance similar to this screenshot. Scroll down until you see the download links. Click the links to download the FASTA files of the chromosomes you need. Make sure you remember to include the mitochondrial and sex chromosomes if they are relevant. As for downloading files via command line, instructions can be found at the link shown on screen. An introductory guide to command line has been linked in the video description. Now that we have gone through the downloading process, let us go over what exactly it is that was downloaded. The FASTA file format is a format created for storing sequence information. The entries stored can be as short as a few nucleotides, in the case of primers and restriction sites, or they can be as long as hundreds of millions of nucleotides, in the case of chromosomes. Each entry consists of two parts: the ID line and the sequence, written across an unspecified number of lines. Therefore, unlike FASTQ files, which strictly follow the rule of 4 lines per entry, in FASTA files, there is no set number of lines per entry. Let's talk a bit more about the ID line in FASTA files. ID lines are lines in the file which begin with a greater than symbol, followed immediately by the name or ID of the entry. Additional information and annotations may also be stored on this line, separated from the ID by a whitespace character. All lines between that ID line and the next ID line are regarded as containing sequence information. With genomic sequences, this will generally be a sequence of A's, C's, G's, and T's, with unknown nucleotides being represented by N's. The sequence itself is typically in uppercase. Nucleotides can be soft masked by being recorded in lowercase, or they can be hard masked by being replaced with N's. There are a multitude of reasons for soft masking or hard masking data, but we won't go into that here. Let us go back to the chromosomal FASTA files we downloaded. They contain one chromosome per FASTA file, so they can be referred to as chromosomal FASTA files. Most chromosomal IDs are given using Arabic numerals, although some organisms, like C. elegans, use Roman numerals. Now that we know what we are working with, we can talk about the modification and combination step. Sometimes, the data in the FASTA files need to be modified before it is suitable for your analysis. This might be something as surface level as changing chromosome names, or it could involve changes to the sequences themselves. Whether or not the information in the FASTA files is modified It is strongly recommended that all relevant files are concatenated into one file containing the sequences for all the chromosomes, also referred to as a genome FASTA file. Depending on the experiment, you may or may not wish to include mitochondrial chromosomes, X and Y chromosomes, and or the genomic sequences of potential experimental artifacts as pseudochromosomes. Making modifications and edits to the chromosomal data is usually unnecessary for well-established organisms such as mice and humans. For cases where modifications and edits are warranted, there are simply too many to be comprehensively covered. When it comes to renaming chromosomes, this can usually be done by opening the file in a minimalist text editor like Notepad or TextEdit, and then directly editing the file. Alternatively, command line can be used. It is also possible to use these text editors to combine files into one for the combination step, but it is recommended to either use a command in command line or a simple self-made script. For the first option, please refer to the command line guide, while the second option requires rudimentary programming ability. After the modification and combination step, we now have a single FASTA file which contains all the chromosomes we need for our reference genome index. Indexing is done to exponentially speed up the alignment process. This is because it is much quicker to look something up in an index than it is to trawl through the data. Therefore, the alignment step in the core pipeline actually aligns the reads against the index and not the genome FASTA file itself. How an index often works is that all subsequences of length k, also known as k-mers, as well as the positions of these subsequences, are recorded. When a read is aligned against an index reference, a k-mer is derived from the first k bases in the sequence. Afterwards, rather than comparing the query sequence against the entire reference, only the k-mer locations are examined in closer detail. The program may still have trouble aligning reads when there are thousands of instances of the same k-mer in the reference genome. If this occurs, it will be noted in the alignment results. Once the indexing is complete, we would have a series of index files with names based on the genome FASTA file. Collectively, these files comprise the index against which the alignment happens.